So as we continue our study of the dogma of molecular biology, we need to look at the details now, again, general details, of translation. So now we're going to look at the process of moving from from RNA to protein. So the language of nucleic acids in the sequence of nucleotides, how do we take that information and turn it into a sequence of amino acids, the language of proteins? So again, it's a triplicate code, right? So we know the genetic code. Any 3 nucleotides you give me, I can tell you what they code for, what amino acid, what of the 20 amino acids that all life uses, what is it dictating is going to go next in the protein. So translation, RNA to protein. So this now again there's a short cartoon video on translation on the website. I want you to go watch that. Watch it. We're going to go over the details, and I'm going to ask you to watch it again. And again, this is the best way to get this stuff in your head. Now, translation's a little bit more complicated than transcription. Instead of just copying into DNA to RNA, instead of copying the language of nucleotides, we're actually going to translate into a whole different language. So we're going to have some more players there. So first of all, we're going to need a ribosome. And we've talked about ribosomes before as the site of protein synthesis. They have a large subunit and a small subunit, and the ribosome is actually composed of ribosomal RNA, rRNA, and proteins. So they make up this big catalytic unit. All right. Now, the other thing that we're going to need is obviously the mRNA that we're going to read the information out of. We're going to need ATP because it costs energy to do this. We're going to need amino acids to build a new protein, and we're going to need the very special molecule called tRNA. And tRNA stands for transfer RNA, but you can also think of it as the translator RNA, because it's going to be the link between the codon in the mRNA and the amino acid adding to the growing polypeptide chain. So the cool thing about nucleic acids, and again Watson-Crick base pairing, is that this mRNA molecule is actually going to Watson-Crick base pair to itself. It's going to fold back on itself into a very specific 3-dimensional structure. On one end of that structure, you're going to have an attachment site for the amino acid. And on the other end of the structure, you're going to have what's called an anticodon. So you can see these different representations. So in the cartoons, you see kind of this kind of columnar structure with the anticodon on one end, but in reality it's kind of this L-shaped molecule as the RNA folds back and Watson-Crick base pairs with itself, complementary base pairs with itself, with the anticodon on the end. That anticodon is very, very specific to the 3 complementary nucleotides in the mRNA. For every codon, there is an anticodon. Therefore, there must be at least as many different tRNAs in a cell as there are codons. And we've already talked about the fact that if you have 4 different letters, you can combine them into combinations of 3, 64 different ways. Now, there aren't quite that many tRNAs in a cell, but you can think of it that way because for every codon, there is an anticodon complement. So ultimately, what we're going to see is those anticodons bind through complementary base pairing to the codon on the mRNA, and within the ribosome, that's going to position then the specific amino acid associated with that specific tRNA to be bound and built onto the growing polypeptide chain. So for example, you see here this tRNA coming in on the right. The anticodon is CCG. So that tRNA with CCG will only ever have glycine attached to it because GGC is the codon for glycine. And you can go back and check that on your chart. So this is the only tRNA that will bind glycine. Right? This tRNA will only bind glycine, and therefore you have fidelity with your process. So GGC then will only bind to CCG as an anticodon, bringing only glycine in to add to the growing polypeptide chain. So let's take a look and break down this process. And once we get going, you can see how it's repetitive and not that hard to understand. So we're going to start with the mRNA out in the cytoplasm, whether that's prokaryotic or eukaryotic. And the start codon is always AUG, and that's always methionine. That's the one exception to the degenerate code. So there's only one codon that encodes for methionine. That's special, AUG. So this tRNA with methionine is going to bind to it, and that's going to allow the whole ribosome to assemble. And now we can start the process. So the ribosome has two sites in it, okay, that are going to have two tRNAs. One tRNA is going to be bound to the growing polypeptide chain, and then the next tRNA is going to be the next amino acid in line. Just when we look at the mRNA, we can see the two adjacent codons. All right. So the process starts with this. In the process of this growing polypeptide chain, we call the P site, call that the peptide site for now, and the A site for the amino acid site, bringing in the next amino acid. So when we have the A site open, there's only the tRNAs that are complementary to the codon present at the bottom of the A site can bind there and stay there. And once they're stuck there, the ribosome catalyzes the formation of a peptide bond in step 2 between the two adjacent amino acids. And then it releases, it kicks out the first tRNA in the first slot in the P site, and the whole thing moves forward one. And as it moves forward one, then it moves that bound tRNA with the growing polypeptide chain into the P site, opening up the A site, waiting for the next tRNA to bring in the next amino acid. Which tRNA is that? It's the only tRNA that is complementary to the next codon, the next 3 nucleotides. And so hence we can get the information in that mRNA molecule to code for the primary structure of a protein. It tells you what order to line up the different amino acids in. Ultimately, when it reaches the stop codon, it'll get kicked off, and now you'll release the polypeptide chain for it to fold and then hopefully become functional. So this is a good time now to go back and watch that brief movie on translation, understanding the components better. Now, here is a summary slide that shows you transcription and translation in a eukaryotic cell. This is a great slide to kind of go back and review to see if you really understand, or ultimately to reproduce on a blank piece of paper. If you can draw this from memory, you're probably in pretty good shape.